The genetic information for all life on Earth is coded in DNA. In all living things, this genetic information is turned on and off as needed in order to express the necessary traits to sustain life. Life forms have been changing the way that their genes are expressed ever since life evolved. With advances in technology, we are able to not only understand how genes are expressed, but we can now turn genes on and off ourselves. I will now describe to you the method we took to change the genes of our new turf grass, CEED. One of the ways that genes can be turned on so that they are expressed is through the use of a protein called a transcription factor. These transcription factors sit on a particular region of DNA called a promoter, which has a unique sequence of base pairs to which only specific transcription factors can bind. Once a transcription factor attaches to a promoter, a large cascade of processes starts to happen, and the end result of which is the expression of the gene that occurs after the promoter sequence. To create CEED, we began by extensively researching gene editing techniques that would increase root growth in grasses. We found two recently published papers which gave us a process to start from. The first, written by Gangping She, described how a promoter taken from a rice plant and then inserted into a wheat plant resulted in greater root growth. The second paper, written by Dan Shen, reported the discovery of a transcription factor in wheat that increases root growth as the concentration of that same transcription factor increases. We then looked for a suitable species of turf to edit. We landed upon Zoysia japonica because its natural root systems are already 2 feet deep, and because its genome has been sequenced and is publicly available. We combined these two pieces of information and began to look through the genome of Zoysia japonica to find genes which were similar to those that code for transcription factor and promoter that were used in the other studies. And we found them. The transcription factor is called NAC, and the promoter is called GRP7. After we found these components, we started to design our new turf grass. To do so, we designed a circular strand of DNA called a plasmid that contains the genetic information for both our GRP7 promoter and NAC transcription factor, which can be inserted into the cell nucleus of the original turf grass. Once inside the nucleus, our DNA plasmid will use CRISPR-Cas9 technology to insert the designed promoter and transcription factors genes into Zoysia japonica's genetic code. The specific way in which this works is quite fascinating. A designed guide RNA routes a Cas9 protein to a specifically chosen location in the genome. Once there, Cas9 acts like a pair of scissors and cuts the DNA open. Once the cut has been made to the DNA, the cell will immediately begin the repair process. The preferred way for a cell to do this involves using a template and rebuilding the damaged region of DNA. Knowing this, we have included DNA on either side of our promoter transcription factor DNA that matches Zoysia japonica's pre-existing genetic code. The cut will heal itself with our information, and our design traits will be included in the plant's full genetic code. If the edit works successfully, our newly designed grass will increase its root growth potential to 3.4 feet, allowing for the sequestration of 85% more carbon than the average turf grass from the air we all breathe, all without changing the above-ground appearance.